Hello, everyone. I hope you're doing great. We're almost through this. This lecture deals with chapter 16, and you need to go through chapter 16. I would look at your notes, definitely read through it, but I don't go through everything on chapter 16. You need to just know the basics and the details that go into DNA replication. And we will go into transcription translation a little later here. But today we're going to look at just DNA replication. And this occurs anytime a cell needs to make a copy of itself, which happens quite frequently because we're always making new cells. We're always having to reproduce our DNA. This word, transformation, is a word you need to be familiar with. Frederick Griffith figured out this idea of transformation that, before, now we kind of take it for granted, but at one point we didn't know what was responsible for what you see. We knew that there was some kind of heredity involved, but we didn't know what the molecule was. And in his experiment, he figured out that if you took bacteria that virulent means deadly, you ended up with a dead mouse. If you took avirulent bacteria, which that's where you can have good bacteria, right? The mouse lives. If you took the heat-killed bad bacteria and injected into the mouse died. But if you took the heat-killed bad bacteria and then a benign bacteria and put them in the mouse, you ended up with a dead mouse because basically the good bacteria ended up taking on the bad bacteria into taking the DNA of the dead bad bacteria and putting that into its DNA, and then you have a dead mouse. Transformation is a really cool thing that we usually do in this unit where we take the E. coli and we end up putting genetic material that makes it bacteria antibiotic resistant. So then we have E. coli that's antibiotic resistant. And we use transformation now and this idea that bacteria will take strands of random DNA and put it into its own genome to have bacteria work for us and make insulin and whatever we're really... Oh, the puppy's here, and I'm hoping she doesn't push the camera away, and have it produce whatever we need. And we'll talk more about that. That's going to be in our genetic engineering discussion. All right, so just to get to the basics, nucleotides are made up of DNA, and this should be a review, okay? Remember, nucleotides are the building blocks of DNA. And you need to know, that's why we're in red and bold, this is very basic, that you have a phosphate group, the 5-carbon sugar for DNA, right? Deoxyribose, and a nitrogen base. And the nitrogen base could be adenine, thymine, guanine, or cytosine for DNA. We have a different base for RNA, remember that. So here we are. This is the basic structure of DNA. And remember, you have this classic double helix on the sides, and you guys built this ladder in the first semester unit where we talked about the building blocks of life. But basically, you have your 5-carbon sugar here, you have your phosphate group, and you have your base, okay? So this right here is one nucleotide. They're attached to each other through these phosphate bonds. Okay, and on the inside, we have the bases, the rungs of the ladder. And that's those base pairing rules. A goes with T, G goes with C. So here, this is actually a general bio slide, just so that you know you've seen it, where you have the 5-carbon sugar, the phosphate, and the base. And this, again, is one nucleotide, right? They're bonded by this phosphodiester bond, which is a very strong bond, okay? So then we're going to look at the structure. I know I'm going through this quickly, but you should know this stuff. These are the phosphodiester bonds on the sides of the ladder, the phosphate with the 5-carbon sugar. So this bond right here, then this is a nucleotide, so then we have this bond right here, okay? The inside of the ladder is made up of these very light hydrogen bonds, okay? Now again, this is review, but you definitely need to know it for your test that's about DNA, that this bond especially is this phosphodiester bond that holds the sides of the ladder together, and then these are the light hydrogen bonds that can be broken quite quickly. Remember, the light hydrogen is like two pinkies touching. A covalent bond is like a full-on grabbing of the arm or fist, or it's a very tight bond. All right, so this is an important thing to understand, the direction of DNA. DNA has a direction. We talk about the 5' and the 3' end. And I used to get so confused by this because I'm like, how can there be 5 carbons? Because we number the carbons. When we always, in deoxyribose or ribose, there's always this, it just never made sense because there weren't actually 5 carbons where they were showing that 5th carbon. But if we label this, I want you to look at this. This is very good for you to see. So we always start in the rightmost corner. So this is carbon 1, carbon 2, carbon 3. Remember, where the 2 lines meet are carbons. So carbon 3, this is carbon 4. And then this guy who's always out here on his own is carbon 5. This is the 5' end. The 5' end very simply has the phosphate attached to it. This is what, when we refer to the 3' end, is right here, because this is carbon 3 right here, okay? Having a basic understanding of how we number these carbons can then help you understand when we talk about the direction of DNA. And now you're going to really have to apply this direction, because when we talk about DNA replicating, you need to understand some of these enzymes only work by adding to the 3' end, not the 5' end. So you kind of got to really start to apply your knowledge of the structure of DNA and how it affects the enzymes' actions. Okay, so when we say the 5' end, that would be this end, because this is carbon 5. When we say the 3' end, we say this is carbon 3, this is the 3' end. So it is basically this last trailing carbon, whether it's this one or this one, that determines the direction. Okay? So there you go. There's your 3'. All right. So the nucleotides in DNA are bonded, these backbones, by basically the 3' binding with the 5' end, okay? And we call that phosphodiester bond. Now, in order for the bases to match up and for the 3' to always bond with the 5' of the next, we have to flip it. If you look, it runs this way, and then we say it's anti-parallel. The complementary strand over here runs in the opposite direction. So now this is the 3' end. So let's do, this is the 5 and this is the 3'. And now this other side begins with the 3'. See, there's your 3' end and ends in the 5' end. And here's your 5'. Okay? Now, this you're going to see has a significant consequence when we go to replicate DNA. And we're going to talk about that. All right. Here's a nice bold red deal. So you're going to have to know this. So if you look, they always run anti-parallel. The 5' end always has the phosphate attached. And then you have the 3' end. Okay. The direction of 5' to 3', direction of one strand, runs counter to the 5' to 3' direction of the other strand. This wording is difficult. If you think about what it says, yes, the direction of the 5' to 3' of one strand runs counter to the 5' to 3' of the other strand. But when you see this on a test, it tends to get people. So make sure you understand what is being said when they use these words, because this is just a difficult concept to explain in words a lot of times. So that's why I gave you this in bold, so that you can see how you could explain it. I want you to go through these table talks. You can pause it, and then I'm going to actually start the other video. But what are the nucleotides within DNA composed of? Hopefully you can go back and answer that. That is your nitrogen base, your 5-carbon sugar, and your phosphate. What is meant by the direction anti-parallel regarding the strands that make up DNA? Okay, there's lots of ways you can say this. I would refer to maybe this to answer it. Use your own wording, but this wording would be a great wording to know and understand so you could apply that to your test. In order to produce a single strand of DNA, the nucleotides combine to form what type of bond? Okay, this can always get people because they're thinking, okay, well, does she want the hydrogen bonds or the phosphate bonds? Well, it says a single strand of DNA. That means you're looking at your phosphodiester bond, okay? The 5' carbon of one combining with the 3' carbon of the other, or the phosphate end, which is the 5' combining with the 3' of another.